All right, welcome back. In the second video, we're going to jump into the details of transcription. How do we get messenger RNA from our DNA? We're going to be focusing on transcription in prokaryotes, although eukaryotes are very similar. Where transcription is occurring, if I look at the DNA double helix, the DNA double helix has to unwind in that region, and that unwound portion is known as the transcription bubble. Transcription always proceeds from the same DNA strand for a particular gene, and that's known as the template strand. In this case, this is my template. I can see that the RNA, the sequence of the messenger RNA, is going to be complementary to the template strand. The other strand of DNA is going to be known as the non-template. This is the non-template strand. And it's also sometimes called the coding strand, because interestingly, it's going to have a similar sequence to the messenger RNA molecule, except where instead of having thymines, we're going to see uracils. So if you look at the sequence of the non-template strand and the messenger RNA, it's almost identical, again, except T's are replaced with U's. All right, so that's that one. A couple of other things to know. The first 5' messenger RNA nucleotide, and I can see that's here, is known as the +1 site or the initiation site. So in this case, it's the adenine right there. Anything before that is known as upstream or are known as upstream nucleotides. So it would be something like over here. If there was more DNA over here, those would be called upstream nucleotides. Anything down past that part are known as downstream nucleotides. And notice that the messenger RNA direction of synthesis is just like DNA replication. It goes from 5' to 3', where you're adding on to the 3' end. Because even messenger RNA and the template strand are anti-parallel, you're going to be starting at the 3' end of the template strand in order to generate your 5' end of messenger RNA. And you can see you work towards the 5' end of DNA. That will be your 3' end of the messenger RNA. There are 3 basic steps of transcription: initiation, elongation, and termination. So in initiation, we're going to see that there is a region called the promoter that will be upstream of the gene of interest, where RNA polymerase will bind and begin transcription. After initiation, we have elongation, which is what it sounds like, where RNA polymerase will read the DNA and add the complementary RNA nucleotides. Finally, you'll reach termination, and there are 2 types of termination for transcription, something called row-dependent and then row-independent, and we'll get into these later. When we think about differences in prokaryotes versus eukaryotes, remember in prokaryotes like bacteria, there's no nucleus, so there's no nuclear membrane. Everything is happening in the cytoplasm. So transcription occurs to make messenger RNA, and as soon as there's enough messenger RNA, translation can occur before even you've finished your messenger RNA. These can occur simultaneously. So that ribosome will grab onto the messenger RNA and start translating that into your polypeptide sequence. And in eukaryotes, though, let's say if this was in eukaryotes, this would be different because eukaryotes, let me draw a cell, we do have a nucleus. So if this were the nucleus, then we have transcription is going to occur here. Transcription going from DNA to my messenger RNA. Then the messenger RNA has to move to the cytoplasm, and that's where it's going to be read and converted to proteins through the process of translation. So how does RNA polymerase know where to bind and start transcription? It looks for a specific region known as the promoter. And in this picture, this whole part is called the promoter. That's where RNA polymerase will bind so that it knows where to begin transcription. And the promoter is always upstream of our gene of interest. So let's jump back to the first step of transcription, which is known as initiation. It turns out that upstream of that initiation site, there are specific regions known as consensus sequences. And these are in a region known as the promoter. So promoters are upstream of the transcription start site, and within the promoter there are two consensus sequences that are similar across all promoters and across most bacterial species. So in particular, there's a region called the -10, so -10 nucleotides upstream of the transcription initiation site. This is called the -10 region, and we can see it says TAAT. And upstream of that, there's another region called the -35 region that has a specific sequence. Again, these two sequences, don't worry about memorizing them, but these are consensus sequences that we see in different species of bacteria. And we also see them in all promoters for these bacteria. If I look at the RNA polymerase, it's a huge enzyme. So it's that yellow or creamish color enzyme, and it's made up of 4 subunits. In our class, I don't require you to memorize them, but there are several subunits that make up this RNA polymerase. I would like you to know the 5th subunit, which is the sigma factor. So that's another subunit of the RNA polymerase. And more importantly, it's the one that recognizes the consensus sequence found in the promoter. So it helps us localize where we're going to start the transcription. So when we combine the 4 subunits of RNA polymerase with that sigma factor, we get the whole RNA polymerase complete enzyme, and that is known as the holoenzyme. After initiation, we have elongation, and here the RNA polymerase just keeps moving down the template strand of DNA. This process starts with the release of the sigma subunit from the RNA polymerase, and that's why you don't see that subunit that we saw in the previous picture. The messenger RNA, again, is synthesized in the 5' to 3' direction as it unwinds the DNA double helix. So you'll see another bubble open up here, and the previous region will close up once we no longer need to generate messenger RNA in that region because it's already complete. So if we read A, for example, we would put down U, T would be put down A, C, etc. And this continues in that 5' to 3' direction. And in prokaryotes, this is very fast. So this is a rate in prokaryotes like bacteria. 40 nucleotides per second. Extremely rapid. So we had initiation, elongation, and our last step is termination. And we have 2 types of termination. One is called row-dependent termination. And in row-dependent termination, we're going to see that there is a row protein that tracks behind the RNA polymerase and is going to bump into everything and allow it to fall apart. In row-independent termination, we're going to see that there are specific sequences in the DNA template strand that is going to cause a weird shape to be formed in messenger RNA called a hairpin, and then everything will break apart. So again, it's either going to be due to a row protein, so that one is associated with this one, or the formation of a hairpin structure, which is associated with this one. And both of these cause the release of the messenger RNA strand so that it can start the process of translation. Here are some pictures of this process. On the right, we have the row-dependent termination, and I can see what's happening here is this is my DNA. This is my messenger RNA in red. So this is my DNA, the template strand. This is my messenger RNA in red. So row protein threads itself across the messenger RNA and pushes everything apart, so everything falls off. And I have my messenger RNA ready to go. And in row-independent termination, which is shown on the left, this is row-independent. We're going to see that there is a specific sequence of nucleotides in the DNA that causes the formation of a hairpin loop in my messenger RNA. So that's a hairpin loop. Immediately following the hairpin loop in the DNA, you have a bunch of A's. That's my terminator sequence. And of course, the complementary nucleotide are uracils in the messenger RNA. Because remember, A and U only pair with 2 hydrogen bonds, it's weaker than G's and C's, which pair with 3 hydrogen bonds. This hairpin loop, in combination with the weaker hydrogen bonds between the messenger RNA and DNA, cause the structure to fall apart. So that releases the messenger RNA. And again, it's really because of that really bulky hairpin loop and the weak interaction between A's and U's. Remember that prokaryotes like bacteria, since they do not have a nucleus, as soon as they create their messenger RNA through transcription, they can immediately translate that messenger RNA into proteins. And that's what's shown in this picture from our book, although I don't think it's the best picture. It's hard to see what's going on. In our book, this is DNA. The green line is supposed to be DNA. And then it looks like it's not shown, but we have RNA polymerases that made the blue squiggly lines, which are our messenger RNAs. And at the same time, we have ribosomes, the red, that are bound to the messenger RNA and they're going to be starting the translation process. If you have multiple ribosomes on a messenger RNA molecule, we call that a polyribosome. So really the take-home message of this picture is that in bacteria or other prokaryotes like archaea, we can have transcription and translation. I'm just going to abbreviate translation happening at the same time. Everything's happening in the cytoplasm. And this will not be true in eukaryotes because we have a nucleus. So this is a picture I found on the internet. I thought it was much better, but there actually was an error in the 5 to 3' end, so I fixed it, and here I'm giving it to you guys. It looks like I'm going to start from here on the left. This is my DNA, double-stranded DNA, and I have RNA polymerase transcribing the DNA. This looks like the template strand is the top one. That's the template strand. I always read the template strand from 3 to 5 in order to make messenger RNA go from 5 to 3. And then we're zooming in here. And this is again in prokaryotes because we don't have a nucleus. We want to see how things happen at the same time, how transcription and translation can happen at the same time. So I'm just taking that template strand, this top strand. This would be the 5' end of my DNA. And I'm making my messenger RNA. So this would be the oldest one. I transcribed this one first. And I can tell because it's the longest one. Then this one is the second longest, third. This is the newest messenger RNA in pink, that pink line that has been formed. So this RNA polymerase latched on first, then this one, then this one, then this one. So I can see that as soon as I start transcription with my first RNA polymerase, as soon as there's space, the next ones can come on and make more messenger RNA. And then my ribosomes, which are these structures, can bind to the 5' end of messenger RNA and start making my polypeptide, my amino acid chains. And the ribosome is moving from 5 to 3' on the messenger RNA molecule. So I can see that's the direction of translation. All right, similar to what you did at the end of the first video, there's an activity in Canvas, another page after this video where I'd like you to watch this YouTube video. I was going to try to embed it, but I learned from the previous lecture, the previous chapter, that doesn't work well in my videos. So I'm going to post the link on our Canvas page. Please watch the video. And again, it's also available through our OpenStax textbook. This is a good one to review the transcription process, going through initiation, elongation, and termination. And that takes us to the end of our second video. In our third video, we're going to look at differences between prokaryotic and eukaryotic transcription.